DNA replication is the process by which we make more DNA. We always need to make more DNA, especially when our cells are dividing, because every single time a cell divides, each of the new cells needs a copy of the DNA. So I'm going to draw a simplified version of how DNA replication occurs. So I'm not going to draw the double helix, or I'm not going to draw the double helix, rather, just because I think it'll make it a little bit more confusing to follow along. So I'm just going to draw the double strands of DNA molecule here. And let's just make up a sequence. We've got A, T, C, C, G. And we know, based on base pairing rules, that A bonds with T, T bonds with A, C bonds with G, G here, and C here. So this would be our double strand of DNA. And keep in mind that those two strands are held together by hydrogen bonds. In fact, I'll go ahead and draw those. Remember, those are hydrogen bonds. Now, it's very important that we have hydrogen bonds there. If you recall from when we did talk about water, hydrogen bonds are weak, and we need those bonds to be weak so that those two strands can separate. So the first step in DNA replication is the separation of those hydrogen bonds, so that now you have two single strands of DNA. You've got your A, T, C, C, G over here, and your T, A, G, G, C over here. Now they are completely separate and are ready to get a new partner, if you will. So these two strands are now separated and they're not going to come back together. What's going to happen is new nucleotides. So let's say there's a nucleotide floating around. It has a cytosine, another one has a guanine, another one has a thiamine, and these nucleotides are actually going to come in and pair up with these available bases. So now you've got cytosine here, you've got guanine here, you'll have another guanine over here, an adenine here, and a thiamine here. The same thing is true for this strand. You're going to have free nucleotides coming in to bond with these nitrogen bases over here. So C will bond with G, again C bonds with G, T bonds with A, and A bonds with T. So ultimately what you have is this kind of fractured backbone of nucleotides that have bonded to your original template. So this was the original strand and this was the original strand. They were templates. They provided the instruction for making a new strand of DNA. Now, we still have a problem because the only thing that's connecting these two nitrogen bases are some hydrogen bonds. So we have an enzyme that actually comes along and connects those backbones so that you have a nice solid backbone all the way down. And now you end up having two molecules of DNA. Notice that both of these molecules, if you're just looking at the sequence, are identical to the original molecule. You had AT, AT, TA, CG, CG, GC. They are identical, as is this one. And the reason for that is that both of these orange strands right here, the original strands, they acted as templates for making new DNA. Now, one thing you really need to notice is that all the new molecules each have one old strand and one new strand. So this one right here is the old strand. It came from the original molecule. And then the new strand was the one that we had made, right? So the green and purple here, that's the new strand that was made through DNA replication. The fact that every molecule of DNA is made up of one old strand, one new strand, means that the process of DNA replication is semi-conservative. And that means that a part of the old strand or the old molecule of DNA is saved. Conserve means save. Part of that old molecule was saved. Now, anytime I ask you to give me the complementary strand of DNA, I might give you a sequence. A, G, G, A, C, T, A, T, G. You have to give me the complementary strand. So what would the nitrogen bases be that bond to each of these? You would have T, C, C, T, G, A, T, T, A, C. And the way you figure that out is based on that Chargaff's rule that we talked about in the previous video. A always bonds with T, C always bonds with G. That's the rule that you use in order to tell me the complementary strand. Complementary. Okay, so these two strands are complementary to each other. They make up a whole. Okay, now you need to be able to do this so that once we start talking about protein synthesis, you can catch on fairly quickly, but this has to come first. Make sure you're comfortable with this, and we'll do a little bit of practice in class as well.